In this session, we are going to learn about the chemiosmotic hypothesis that explains how the ATP is formed during the light reaction of photosynthesis. So, according to the chemiosmotic theory, the ATP formation is because of generation of proton gradient, proton or H plus gradient, and then there will be a breakdown of this gradient that will lead to the formation of ATP. So first of all, there will be creation of a proton or H plus gradient inside the lumen of thylakoid, and then there will be a breakdown of that proton gradient that will cause the production of ATP. So this is basically the chemiosmotic theory, which explains how the ATP production occurs. So let us understand it in detail with the help of the diagram. So let me draw a thylakoid. So this is a thylakoid. We are talking about light reaction here. So obviously it will be occurring in it will be associated with thylakoid, right? The membrane of the thylakoid. So first of all, there will be a creation of a proton gradient. Now let us understand how this proton gradient will be created. So we studied about the different photosystems and the electron transport chain in a previous lecture that occurs, or the important systems which are involved in the light reaction. So there's photosystem 1, photosystem 2, then there's photosystem 1, and in between these two systems there's electron transport chain, right? Suppose this is photosystem 1. Here there is photosystem 2, and in between there is electron transport chain. So in the electron transport chain, I'll draw the 3 electron acceptors: plastoquinone, then cytochrome B6F, and then plastocyanin. So the electrons will flow from here to here and then to photosystem 1. Alright, so we studied that in photosystem 2 there's an event called photolysis of water or splitting of water. So what happens in the splitting of water? The water molecule is broken down into H plus ion, electrons, and oxygen gas is released. So this H plus ion, which is produced during the photolysis of water, this enters the lumen of the thylakoid. Alright, now when the electrons pass from photosystem 2 to photosystem 1 through the electron transport chain, then when the electron is passed from plastoquinone to cytochrome B6F, during this flow of electron, one hydrogen ion enters inside the lumen here. So this hydrogen ion will enter inside the lumen. So we can see that how there's accumulation of proton or H plus ion inside the lumen of the thylakoid, right? These hydrogen ions will keep entering the lumen in this way, first through the photolysis of water, and also when the electrons are flowing through the electron transport system. Now since this hydrogen ion which entered through the electron transport system, it is coming from the stroma of the chloroplast, right? Outside the thylakoid there will be stroma. We studied about the structure of chloroplast. So this is stroma. This is also stroma. So inside the stroma there will be deficiency of hydrogen ions or protons, because these protons are entering through the electron transport system inside the lumen of thylakoid. Alright, also if you remember, in the PS1, in the photosystem 1, there is production of NADPH. NADPH, right? So the production of NADPH also requires a proton or H plus ion. So this H plus ion combines with NADP plus and forms NADPH. So one hydrogen ion or proton is also taken up from the stroma. So you can clearly see that the hydrogen's ions are continuously being picked up from the stroma, whether it is during the formation of NADPH or whether it is during the flow of electron during the electron transport system. So that causes a deficiency of hydrogen ions in the stroma, or decrease in the number of hydrogen ion inside the stroma. But if we look at the lumen of the thylakoid, that the lumen of the thylakoid is accumulating more and more hydrogen ions or protons, right? The proton is coming from the photolysis of water as well as through the electron flow in the electron transport chain. So this is creating a proton gradient. Now what do you mean by proton gradient? It means that the concentration of hydrogen ion is more inside the lumen of the thylakoid as compared to the concentration of hydrogen ion in the stroma. So there is less number of hydrogen ion in the stroma and more number of hydrogen ion in the lumen. So this causes the pH of the lumen to be low. Low pH means it is acidic because of more number of H plus ions here. So we can see that a proton gradient has been established now. A proton gradient has been created. Alright, so this is how the proton gradient is created inside the chloroplast. Now we need to understand how this proton gradient will be broken down so that ATP is produced. So for the breakdown of this electron gradient, what happens? There's a ATP synthase enzyme which is present in the membrane of thylakoid here. It has two parts. Alright, so let me write it down first. So there will be an enzyme called ATP synthase which will synthesize ATP during the breakdown of proton gradient. Now this has two parts. This enzyme has two parts. So the first part is called CF0 and the second part is called CF1. Alright, so this CF0 is a transmembrane protein channel. It is embedded inside the membrane of the thylakoid. So if we look at here, I can draw a transmembrane channel like this, which is embedded inside the membrane of thylakoid. So this is one part of the ATP synthase enzyme, CF0, and then there's another part which is the CF1. It protrudes out from the membrane. and it faces the stroma. So this part of the enzyme faces the stroma and is situated towards the outside of the membrane. So let me draw it here like this. So this is CF1 part of the ATP synthase enzyme. It is also called as F1F0 complex. So this forms the ATP synthase enzyme. Now what will happen, because of the accumulation of H plus ions inside the lumen, because of the creation of the proton gradient, this H plus ion will need to be pumped out, not pumped out, but these H plus ions will need to be thrown out of the lumen to establish equilibrium, to maintain the equilibrium of this proton balance inside the lumen and outside the lumen, because outside the lumen there is stroma where there is deficiency of H plus ion. So now to maintain this equilibrium, these H plus ions will now pass out. They will be transported out through this F1 F0 complex. Alright, so this H plus ion will now, they will be transported out of this F1 F0 complex of ATP synthase enzyme and they will enter the stroma. Now this passage of H plus ion through the F1 F0 complex of ATP synthase is a highly energetic process. It releases a large amount of energy which activates the ATP synthase enzyme present in the CF1 part. So this CF1 part has ATP synthase enzyme. Suppose this is the ATP synthase present inside the CF1. Now when the hydrogen ions pass through this complex, a high amount of energy is released which activates this ATP synthase enzyme. And when this ATP synthase enzyme is activated, then ATP is produced. So during the passage of this H plus ion, there will be production of ATP from ADP. Alright, so this is how the ATP is produced according to the chemiosmotic hypothesis. I hope it is clear to you. I will summarize it again in short for you. So what happens? The chemiosmotic theory or hypothesis. According to the chemiosmotic hypothesis, the ATP production during the light reaction of photosynthesis is because of this theory basically explains how and why the ATP synthesis occurs during the light reaction of photosynthesis, and it is based on the fact that first of all there will be generation of a proton gradient, and then there will be a breakdown of that proton gradient that will cause the ATP synthesis to occur. Alright, so how the proton gradient is generated? So first of all, we understood how this proton gradient is established in the thylakoid lumen and the stroma. The hydrogen ions are continuously taken up from the stroma during the formation of NADPH and during the electron transport chain. And then these ions, hydrogen ions, are also continuously accumulating inside the lumen of the thylakoid during the photolysis of water, which is associated with photosystem 2, and during the movement of electrons while it passes through the electron transport chain. Hydrogen ions are also entering inside the lumen from there. So that causes an increase in the concentration of hydrogen ion inside the lumen, causing the pH to be low or acidic. And in the stroma, there is decrease in the concentration of ion. So this imbalance in the concentration creates the proton gradient. Now we need to break this proton gradient to establish an equilibrium, right? So for that, the ATP synthase enzyme comes into picture. This enzyme has two parts, the CF knot, which is a transmembrane channel, and it is embedded inside the membrane of the thylakoid here, and the other part protrudes out of the membrane, which is the CF1, and it has the ATP synthase enzyme. CF1 faces the stroma. Now this protein complex F1 F0 complex will help in breaking down the proton gradient. How it helps? It provides a passage for the H plus ions to be transported out of the lumen to the stroma. And when the hydrogen ions pass through this protein complex, through the F1 F0 complex of ATP synthase, high amount of energy is released. That energy is sufficient to activate the ATP synthase enzyme. Now this enzyme consumes this energy, becomes activated, and it converts ATP to ADP to ATP, and that is how the ATP is formed during the light reaction of photosynthesis. Alright, so I hope it is clear to you now that what is chemiosmotic hypothesis and how the ATP is formed according to the chemiosmotic hypothesis. So this in a way concludes that how the ATP and NADPH are produced in the light reaction. If you remember, when we started the lecture, or specifically in the light reaction, we studied that the two main products of light reaction are NADPH and ATP. So NADPH was already produced when we studied about the electron transport chain. Right, in the photosystem 1, the NADPH was produced, which we also see here, which we also saw here in the case of this proton gradient creation. So NADPH was already formed. Now we know that how the ATP is already formed, is also formed during the light reaction of photosynthesis, right? So now it is clear that in the light reaction, these NADPH and ATP is formed, and how they are formed and where specifically they are formed. Alright, so this was all for this lecture. Rest other parts we will continue in the next lecture. Thank you so much.